joined UC Cooperative Extension as an integrated pest management advisor at the beginning of June 22. Although he's based in San Diego, Eric is also responsible for the integrated pest management needs in Orange and Los Angeles County. His background is in entomology, and he focused primarily on managing arthropod pests. Eric works with growers and communities wherever pests are an issue, including in nurseries, floriculture production, citrus, and avocado crops, and the many small farms found in Southern California. Eric, if I remember correct, we met you sometime that was a month to a month and a half ago. We were at Ophelia's farm, and you were presenting about the work that you're doing on pests in agave. I think this is a very timely issue. These issues are growing. So without further ado, I really want to thank you for being the first speaking on our series. Welcome.
Thank you. Appreciate it. All right. So yeah, I'm going to be talking about one specific pest of agave, talking about identifying and managing agave mites. And there's a whole lot of information for this. If you've seen any of the previous talks I've given on this, this is kind of a longer and more in-depth version of it. So as an outline of all the different things I'm going to be covering today, first we'll do just a little bit of an overview about agave mites overall, and there's some of the agave mites pictured right there. Then we'll be talking about some of the current research that I'm conducting here at UC Cooperative Extension along with some of my colleagues. And that's looking at the biology of the mites, how to find them, looking into biological control and different miticides, different ways to control these. And then we'll finish talking a bit more specifically about agave mites and the impact that they have for agave grown for spirits. Because I originally started doing this work on agave that are grown for the ornamental industry. So those in nurseries and those being put out in landscapes. But there's a lot of crossover between agave grown for spirits production as well. So let's start with that agave mite overview. So agave mites, what was kind of known so far before we started this research? When it comes to identification, again, here's a picture of some of those agave mites on the right-hand side. Agave mites are colloquially called grease mites. The genus is Oziella, but the species is unknown. And that kind of speaks to the fact that these aren't very well studied at all. Very, very little research has been done on these mites. Unlike a lot of other mites we might be more familiar with, like, say, spider mites or things like that, agave mites are a type of areophoid mite, and that means that they look quite different. They're more tube-like, and they have 4 legs at the front of their body instead of the sort of more normal 8 that we associate with mites. And they're very, very small. They're about 1/3 of a millimeter long, and they're this kind of translucent whitish color. And they're very, very hard to see and find for a number of different reasons, which we'll go into. But of course, their small size and their translucent nature being one of them. But just to give you a sense of how small they are, here is an agave mite on this penny. And you can't see it because it's so small. So if we zoom in on the 20 and the date here of 2022, inside right there, that's how big agave mites are. So they are very, very small. Even under ideal circumstances, they are quite difficult to see. Now, when it comes to their biology and life cycle, these agave mites live and feed on the surface of leaves, but they do so at the very base of the leaves or inside the core of the plant. So if you see this picture of the leaf on the right-hand side, normally you only see the sort of dark green section of the leaf, and then at the bottom of that whitest section, you'd only see that if you rip the leaf out of the plant. That whitest section is where the agave mites live and feed. So even if you had really good magnification and you were looking at this agave, you wouldn't even see the mites because you have to rip apart the plant in order to find them because they live sort of deep down on the surface of the leaves. And the damage they cause is by their feeding occurs on the surface of the leaves. It's unseen at first, and then as the agave grows, the damage starts to grow out. And then you start to actually see, oh, wait, I might have a problem. I might have these agave mites. Now, they're host specific to agave as far as we're aware. And they can be found on things like blue glow agave, Perry's agave. There's a number of different others. There's no full host list, although this is something that me and a number of my colleagues here at UC Cooperative Extension are now working on as part of a different project to try to better quantify the host range of these agave mites. But there is no full host list currently. As of right now, this is a non-comprehensive host list. This is taken from the literature as well as some personal observation and observation by different growers. So there's a whole bunch of different species. I'm, of course, not going to go through all of these, but you can see a lot of different species are impacted. And one of the sort of overarching common themes seems to be that the species that are impacted and the different varieties that are impacted are those with a kind of powdery blue leaf color. and also medium to semi-large leaves. So they don't have like the really, really narrow leaf varieties, and they're not the really big, broad leaf varieties. It's kind of like middling leaf size and usually a kind of powdery bluish appearance. Those seem to be the ones that are most likely to be impacted by agave mites. So what does their damage look like? I told you they were colloquially called grease mites, and here's why. The damage looks kind of like greasy streaks. So it looks like someone dipped their finger in grease and then smeared it over the surface of the agave leaf. And you'll see that on those sections where it's grown out, the sort of more, powdery blue, greenish sections. But then also, as the feeding damage progresses, you'll start to see lesions like you can see on this leaf here, down in the more whitish section of the leaf. Additionally, damage can look like yellowing leaves. So here, not only can you see kind of greasy sections on the core and deep towards the interior of the plant, but it's also yellowing out a little bit. As the damage progresses, eventually the core will start collapsing. It'll start getting all twisted, distorted. You can see it's all greasy and all covered in lesions here, and it really can start to impact the growth of the agave as well. So these are really kind of the common signs or symptoms, of agave mite infestations. Now, this damage is rarely fatal. It can kill the plants. It definitely can kill them, but usually it doesn't. But even if it doesn't kill the plant, it certainly slows down the growth very significantly. And then from an ornamental perspective, it definitely harms their appearance. For spirits agave, this also can be problematic because if your agave is growing even more slowly than normal, well, that's also going to be an issue for you. It's going to take a lot longer for you to reach an appropriate size for harvest as well. Here's a bunch of different pictures on what it looks like. This is agave Americana, I believe, where you can see kind of the greasy sections, some of the lesions closer down in towards the base of the leaves. This one's a little bit more subtle, but you can see greasy sections on some of the leaves and also how the core isn't growing particularly well. It's definitely a little stunned. The leaves are looking a little strange. Here's some more pictures where the damage is very severe. These are on blue glow agave, where the leaves are all sort of torn. They've kind of fallen apart because there's so much damage to them. The center is all completely greasy, yellow, stunted. That's a very, very severe case of damage. Here's another example where you can see sort of a more clean blue glow at the bottom and then a much more mite-infested one in the middle, where again, collapsing core, greasy sections, yellowing leaves, very indicative of agave mite damage. Here's some examples from agave that are grown for agave spirits production. So these are agave thuringiensis, and you can see the sort of greasy lesions leading up the leaves. Core isn't doing particularly well. Here's another example where you can see the grease lower down on the leaves, and the core, again, is kind of twisted and not growing particularly well either. Here's some of them that were planted out in the field. We'll talk a little bit more about this at the very end of the presentation, but they look terrible. You can see that they're not growing well at all. These things are probably not going to survive, or even if they do, they're going to be extremely stunted for their entire life. And here's one more example. Twisted core, greasy sections, so clearly not doing particularly well. All right, so that's what the damage looks like. What about in terms of management? What was known about management before we started our research? And basically what it came down to was that it was quite difficult. And that's because the mites are protected at the base of the leaves and inside the core of the plant. It's really hard to get anything to reach them, even if you are applying different miticides. Additionally, the symptoms appear well after the infestation has taken place. People didn't really know exactly how long, but it was clear that the mites had been there for a long time before you even start to see symptoms. And therefore, your plants are probably infested for months or weeks before you even notice that anything is wrong. So all this makes it quite difficult to try to manage these mites. What you want to be doing is looking out for signs and symptoms like this, where, oh, the plant is starting to show symptoms. You want to be on the lookout for those as soon as possible, spot them, and if you do, ideally, you'd want to be culling these infested plants. So get rid of them. The damage is not going to heal itself very quickly. They can recover, but it usually takes a while, and there's no guarantee of that if you don't have any additional treatment put on them. If you do cull the plants, cover them and keep them downwind of your other plants, because agave mites, like other areophoid mites, are so light that they can travel on air currents and just kind of float on the wind and get to new places. So be aware of that and be careful if you're culling them. One of the other recommendations, one of the only other recommendations was for getting rid of agave mites, was to core your agave, basically remove the center of it. So you'd take a spade bit, you'd put it on a drill, you'd blast out the center of your agave, and then basically you would hope that pups would form out of the bottom and you'd be able to propagate it from that, and the pups would be clean of the different mites. So that's clearly very extreme. That's not a very good option for lots of different people in lots of different contexts. But that was one of the few sort of options that was actually out there. There was some information, basically all anecdotal, about miticides. Some people saying systemic miticides might work, or miticides combined with adjuvants to really increase the penetration could work well. But again, this was all anecdotal, and it seemed to be mostly ineffective. For the most part, no one was really really having good success of controlling agave mites, which is why one of the suggestions was, coring your agave, like we mentioned before. So very, very little was known about managing agave mites, and it basically came down to kind of extreme measures or just getting rid of your plants. So there was a big dearth of knowledge here. That was the extent of what was known about agave mites, and growers in San Diego that I've been working with and that some of my colleagues have been working with were having this pest in ornamental agave, and we're having a lot of trouble managing it. In some cases, they were getting basically up to 40% yield loss on their blue glow agaves, for example, that they were selling for ornamental production. So that's a really, really big loss for them. So this is quite a big problem. So one of my colleagues, Jerry Spinelli, and I started this research project to determine best management strategies for how do we actually deal with agave mites. And that takes us to the next section, which is the current research that we're doing here at UC Cooperative Extension. Here's Jerry Spinelli, again, one of my colleagues who I started this work with, out in the greenhouse taking a look at some of the plants we were using. So for the current research, there was kind of 4 main areas we were looking at, and we quickly realized that because so little was known about agave mites, we had to do some of that basic research, some of that basic biology of like, how do these mites even work? How do we even work with them? How do you work with them in the lab? How do you, start to do experiments with them? So we did a whole bunch of basic stuff like that to begin with. And then we also did a little bit of work about investigating how infestations occur. So how long does it take from an infestation starting to when symptoms appear, and also do the mites spread really easily? Then we were looking at management options, which is what we really want to do in the first place, which was investigating, say, biological control. Can predators reduce or eliminate agave mite infestations? And then, of course, looking at different miticides, different chemicals that might be effective for killing the mites on currently infested plants, and then also which ones could be used preventatively to keep infestations from occurring. Let's talk about these kind of first 2 sections really quickly together, because there's a lot of information in here and not all of it is relevant to different growers. But first off, when it comes to symptoms and spread, what we found is that it takes about 3 to 5 months after a plant is infested with agave mites for symptoms to appear. So the mites are present, they're feeding on the plant for about 3 to 5 months before you even know anything is wrong. So yeah, they're present for a long time before you start to see the damage. Additionally, mite spread is pretty unpredictable, even when pest pressure is high. So even in situations where there's a whole lot of plants that have agave mites, it still doesn't seem like, all the other plants around them get infested. It's really unpredictable how exactly these are going to work, and we don't know why that's the case. Plants close to infected plants seemed like they were more likely to get the mites, but this definitely wasn't always the case. And sometimes plants far away would get the mites, whereas plants close to an infected one wouldn't. So as an example, we were working in a greenhouse or a high tunnel where there were infested blue glow agave popping up all over the place. So basically the high tunnel was filled with blue glow agave, and infestations were constantly appearing. And so we thought, okay, well, we're going to put 100 clean plants in this high tunnel, plants that we know don't have mites, and we're going to see how long it takes for our plants to start getting these symptoms, basically to kind of get an idea of, how long does it take for ambient mites to come in and infest our plants. We did this for 6 months in an area where infestations were common, and not a single one of our plants showed symptoms or signs of mites the entire time. And this was us cutting open the plants and checking to see if there were mites inside of them. So all this to say that agave mites are both tricky and sneaky and very unpredictable. So it's kind of hard to figure out where exactly they're going to go. Takeaway from all this is that you should be vigilant for any symptoms, and you should check plants, even those far away from an infestation, because you need to act quickly if symptoms are seen, because the mites have been there for a while and have already been causing damage for a little while. One of the other things that we looked into that certainly can be useful for growers or people who are doing this on their own is that it's really hard to find the mites, and you can find them before symptoms show up, but even then you need kind of specialized tools. And that's where we figured out that UV light was a good option. So mites, even under good magnification, and you do need good magnification to see them, even under good magnification are hard to see under normal lighting conditions. Here's a bunch of mites and their eggs under normal lighting conditions. Hard to see because they're translucent, they're tiny, and you have to angle the leaves just so in order for them to catch the light, and so you can actually end up seeing them. Agave mites and their eggs, however, fluoresce weakly when exposed to ultraviolet light, when exposed to very strong ultraviolet light. And so this is what it looks like under normal light. And here's the exact same picture, but now with UV light. And you can see the mites are popping a lot more. Their eggs are popping a lot more. They're far easier to see. So for us, this was really, really useful because we were trying to count how many mites were on plants and things like that. But there is another side of it that's very useful for growers. Note, you do have to use a very powerful flashlight with a filter on it, and we'll talk a little bit about which specific ones we used later on. And also note, even if you have this, and even if you have good magnification, finding mites will still take effort and time. But something we did find that's more useful is that not only do the mites fluoresce under ultraviolet light, but their heavy feeding damage caused by the mites also fluoresces. So lesions that the mites are leaving can be seen with the naked eye when using ultraviolet light. So here's the picture on the left, that's normal light. The picture on the right is with UV light. And this kind of tiny little white stippled section right there, that lighter section, that is indicative of heavy mite feeding. You can see it much better in real life. It doesn't quite show up well in this picture right now. We'll see some better pictures in just a second. But these kind of subtle lesions like this correlate with high mite and egg populations, usually like 50 plus just in this tiny little lesion that you see here. Here's another example of it where the picture on the left is under normal light, the picture on the right is under ultraviolet light, and you can see the lesion much better here, where it was hard to see under normal lighting conditions, but here it's much more apparent. So it really pops out. And again, you don't need magnification to see this. You can see this just with the naked eye and be able to tell, oh, there is actually an active mite infestation and they are causing damage. Here's another two examples right here under ultraviolet light and right here under ultraviolet light, where you can really see those lesions where, again, there's probably hundreds of mites and hundreds of their eggs, and they have an active infestation going. So what different tools can you use to find the mites? Because the goal of having these tools beforehand is that, maybe you want to check some of your agave before they start showing symptoms. If you're suspicious you might have mites, it may be worth it to sacrifice one of your agaves, rip it up, and look for lesions like this. So what tools should you have? Could you be able to use for this? The tools you have, first off, if you want to use ultraviolet light like this, we really highly recommend the Welltool M2BF flashlight. Pictured up here, you can buy it off Amazon, you can buy it online. It's a very good black light. It allows you to see those lesions and allows you to see areas with high infestations. And if you really want to use magnification and look for them as well, then it makes it much easier to find the mites. It's a little expensive. Even though no magnification is needed, it's about $90 for this flashlight. So it's pricey, but it can be a very useful tool if you're willing to sacrifice some of your young agave and sort of make sure, okay, they don't have any mites present on them. But yeah, magnification is still good to use even if you have this black light, even though it's not needed, because you can see those lesions. A couple other inexpensive and accessible options that can be used to magnify the agave mites and hopefully see them a bit better are things like a phone camera scope. So these clip onto your phone, they go right over the camera. They're about $20 to $40 online, so pretty economical. Again, if you look up phone camera microscope, you can find something just like this. So pretty effective. Here's an example of a picture that I took with a phone camera microscope, and you can see there is an agave mite kind of in the center on this lesion right over here. So that works pretty well. You can also use handheld digital scopes. It's about $200 for a decent model. This can connect to your computer or sometimes can connect to your phone. This will be even better. You can see even a little bit closer and actually get those mites well magnified. So again, all of these have their drawbacks. They take some time and effort to use. They're a little bit difficult and they do cost a bit of money, but these are all useful tools that can be used to visualize the mites. Again, as I said, all take some practice, and it's important to note that to use any of these, you have to kill the plant. So the reason why you'd be using any of these tools is if you want to sacrifice a plant before symptoms appear to check and see if agave mites are present. If you don't want to do that, then just wait for symptoms to appear, and unfortunately, you're going to be a little bit behind the curve. But if you do want to try to get ahead of it, any of these tools will be quite useful for finding agave mites. Okay. That's all we're looking at for right now for sort of the basic biology and investigating how infestations occur. Let's talk a little bit about biological control. So does it work to use predators to get rid of these agave mite infestations? We did a number of different experiments with predatory mites. Here's a pictures of 3 different mites that we are looking at. So we have Amblyceus andersoni, Neoceolus californicus, and Amblyceus swerskii. All of these are commercially available, so you can buy these and then release them. And you can see, it's a little hard to tell from each of these pictures, but clasped inside the jaws of each one of these predatory mites is an agave mite in each one of these pictures. So we actually did find that all of these were found to consume agave mites, and they were also found consuming a related species of aloe mites. So yes, they'll eat areophids like this, but then the question is, what about on a larger scale? Do these actually, can they reduce infestations? Can they prevent agave mite infestations from taking place? Will these predatory mites eat enough agave mites to actually make a difference? So we did a number of different experiments on this. First off, we did a curative study. So basically, we took highly symptomatic agave that definitely had agave mites on them, and we ended up putting a total of 800 predator mites per plant on these highly symptomatic plants. Now, that's a lot of predators. That's a very, very heavy dose of them. And we would put another 800 mites on 2 weeks later. So this is a very, very heavy predator dose to see under best case scenarios, will they clean up agave mite infestations? And we found that there was no effect of treatment. So here on the x-axis, we have our different treatments. We have Andersoni, Californicus, and then at the very end, Swirskyii. Those are the 3 different species. And then the control is in blue. And on the y-axis, we have the number of agave mites after we chopped up the plant, after we'd applied all these predator mites and let them sit a little while. And basically, even though it looks like some of these, like say Swirskyii, has significantly fewer agave mites, there was no statistically significant effective treatment. So none of these were statistically significantly different than the control. And even in the best case scenario, there still would have been hundreds of surviving mites, agave mites on the Swirsky application. The same thing, this time we're looking at agave mite egg abundance. So same axes, except we are now looking at the number of surviving agave mite eggs on the plant. And the same thing is true. So not only did we have no effective treatment for getting rid of the adult agave mites, but also there was no effective treatment for getting rid of the eggs of the mites. So predators of all the different kinds we tested did not eliminate agave mite infestations. Okay, but maybe these predators can be used preventatively. What if we apply the predators and they're around to begin with? Does it prevent agave mites from infesting the plant? So we tried this. Basically, we infested clean plants. We put agave mites on plants that didn't have any agave mites on them, and then immediately added the predators, kind of simulating a situation where the predators are already active, they're already hunting. Would they then prevent the infestations from taking place? And here's what we found. The axes are the same, the colors are the same. Of course, this is just now a preventative treatment. What we find here is that Neoseiulus californicus led to significantly fewer mites being found on any of the plants later on. And the same thing was true of the agave mite eggs, where Neoseiulus californicus led to fewer agave mite eggs and fewer mature agave mites. So basically, what this means is that Neoseiulus californicus, based on these experiments, may prevent agave mite infestations if the predators are there before the agave mites show up. Now, all this being said, kind of a summary of all of this. Amblyseius swirskyii, Neoseiulus californicus, and Amblyseius andersoni, which are again all commercially available, all consume agave mites, but none of them cure existing infestations. It seems that Neoseiulus californicus may be effective if used preventatively. However, I do have some reservations about this. The experiments are kind of tricky. There's a lot of variation in how many agave mites end up establishing. I'm sort of worried that this result might be an artifact of the experiment we were running. So I want to run this experiment again to make sure. But as of right now, it seems that Neoseiulus californicus might be effective if used preventatively. I would just hesitate to take that to the bank. I would say, let's wait and get a little bit more data on that. I'd like to run one more experiment and make sure that's true. But for now, those are the results that we have, and they could be useful if used preventatively. All right, last section, we'll talk a little bit about different miticides that were effective against agave mites. So what different chemistries can be used against these mites? When we're talking about miticide trials, we tested a whole bunch of different miticides, and we wanted to focus on ones that killed related aloe mites. So a little bit of research has been done on aloe mites, and some miticides are effective against them. Based on that, we're like, okay, let's try those ones and see if they work against agave mites. So we tested abamectin. These are all the active ingredients we tested. Abamectin, spirotetrafat, spiromezephin, and fenpyroxamate. These are commonly used in the ornamental industry. They're not something that'd be available to spirits growers, but this is a good place to start with to see what even if you are using sort of a full suite of chemistries can be used against agave mite. So we found that the results were very, very disappointing. For both agave mite abundance and agave mite egg abundance, there was no effective treatment. So on the x-axis of all of these, we have the different treatments we were testing. Y-axis is the number of mites on the left-hand side, and then on the right-hand graph, it's the number of eggs, similar to what we saw before. There's no effective treatment. You can see all of these are basically exactly the same as each other. So even when we're applying these miticides, they don't clean up existing agave mite infestations. It seems that the agave mites are protected inside the plant and they are not killed off by these miticides. Okay, we tried some other ones though. Penetration is important, so maybe adding some adjuvants in to try to break the surface tension and really allow those miticides to soak in and get down into the core and at the base of leaves where all those mites are. Maybe those will help. We tried that as well. We tried all of those same things with a non-ionic surfactant added to try to break up that surface tension and get the miticide down lower. And we found the exact same thing. It looks a little bit different here, like maybe abamectin in yellow was actually working, but the same thing is true that there was no statistically significant effective treatment. In all cases, there were surviving agave mites, and again, there was no difference that we could observe between the control and any of these different treatments, even when we added this surfactant to them. Okay, we tried some other compounds. We're doing some IR4 testing, doing a bunch of other compounds to see if anything can try to kill these agave mites once they're already established. We looked at 2 different concentrations of castor oil and peppermint oil, and then we also did an unlabeled compound containing peppermint and clove oils. So these are kind of more natural remedies that have been found to work against areophid mites, and we wanted to test them here and see, well, do they actually work against agave mites? Because they have been effective against other types of areophoid mites. And here, exact same story as normal. It kind of looks like there's some differences. The only thing that's different about this graph is the control is now in a different color, but everything else basically the same. And there was no effective treatment, not on the number of agave mites surviving, nor on the number of agave mite eggs surviving. So what about that? That didn't work at all. So curative miticides weren't working. What about using miticides preventatively? Because this seems like a very similar story to the predators. Curatively, the predators didn't work. Preventatively, maybe they did. So we were going to try some things out. And we wanted to try, because none of the miticides we tested cured existing infestations, we wanted to try a specific type of preventative application because some of them showed a little bit of promise. One of the growers that we were working with had started applying sulfur to their plants. That's what's being applied in the background here. And basically, when they were applying this every week or every 2 weeks, they went from having 40% yield loss on their agave to having basically no agave mites present at all. And so we want to validate this. We want to make sure that, yes, this is actually working. So basically what we did here is we were trying 3 different treatments. The control, the untreated control, is going to be in red. In green is Contos, which is spirotetramat, one of the miticides that we were trying before. And then in blue is sulfur. And basically at day 0 on this graph, we applied Contos and we applied sulfur to a bunch of different plants. So we've only applied the miticides once. And then on day 0, on day 8, on day 15, and on day 22, we applied mites to a subset of those plants. And basically what this is trying to figure out is, do these different things, does Contos or do sulfur protect the plants from having infestations? And how long does that last? So does it work right away? Does it work after 8 days? Does it work after 15 days? Does it work after 22 days? So again, just the one treatment of miticide, does it have a preventative and protective effect? And how long does that last? Okay, so on day 0, basically, as soon as the miticides had dried, we threw a bunch of mites on, and then we came back and we looked at it 3 weeks later to see if the mites had established on the plants. And we found that in the control, as you can see here, a lot of mites established. But in the Contos treatment, almost no mites established in the green. Very, very few of them. So Contos did have a preventative effect and keep the mites from establishing. And then sulfur is in blue, and you can't even see it because no mites established when we applied sulfur. So this seems to be in line with what that grower was finding. When you applied sulfur, none of the mites established. But this was, again, on the same day we applied treatment. What about 8 days later? At this point, Contos appears to have lost its preventative qualities. It's now exactly the same as the control, but sulfur still has absolutely nothing. So for the plants that we applied sulfur to, mites weren't establishing at all, even after 8 days. At 15 days, Contos and control are still statistically the same as each other, and there was one single sulfur plant, and one of their leaves had a few mites on it, but that was it. So again, even up to 2 weeks, it seems like sulfur is still preventing the plants from getting infested with mites. And then finally, at 3 weeks, there's still a statistically significant difference where the control and Contos are the same, and sulfur has a couple mites that are establishing, but for the most part, nothing is happening still, even after 3 weeks. So all this to say, sulfur prevented infestations almost entirely up to 2 weeks and significantly reduces infestations up to 3. I think under real world conditions, sulfur would protect your plants for at least 3 weeks. And you could probably get away, say, applying sulfur every 3 weeks and keeping your mites clean, keeping your plants clean of mites. As we said, it also significantly reduced infestations even after that. We were only able to run this experiment for 3 weeks, but I would imagine the effect would continue at least 1 more week. We're going to be testing another number of other compounds soon to see if they also work. But for right now, sulfur does definitely show some promise and seems to work preventatively. So curing existing infestations with miticides is very hard, even if you're using all the different available miticides that are available to growers anywhere. Prevention is definitely the way to go instead. And what we found is that sulfur works well up to 3 weeks. Again, it works almost perfectly up to 2 weeks and still works well up to 3 weeks. Other options haven't been tested, but they certainly might work. Again, we're going to be testing these shortly, but things like different oils or insecticidal soaps or some other products might work similarly to sulfur, and there may be some other options that we can use other than just sulfur preventatively. Okay. That's been a lot of stuff. Let's finish up with a little bit of information specifically about agave mites in agave that are grown for spirits production. So, agave mites in agave spirits production. This isn't something that I originally done too much work on. Again, I was working mostly with people in the ornamental side of things. But the most common question I would get asked whenever I gave presentations on this was, what about tequiliana? Like, what about the tequila? Is that safe from the agave mites? And I didn't know what the answer was. So this was definitely something where I hadn't heard about mites being present on them, so I assumed it wasn't a host. And spirits agave was a much more niche market, especially here in the United States. Ornamental agave is a far bigger thing. So I hadn't devoted too much attention to it, but it definitely was a little bit interesting. We had a grower who reached out to me about potential agave mite symptoms on their plants. And if you're familiar with Ophelia, she was the grower who we had agave field days with. She was the one who reached out. I was saying I might be having some agave mite issues. So went out, checked those out. She had a whole bunch of different varieties growing. And it was a really good place to gather more information. And this is also important because there's even more unknowns about agave mites and spirits production, and fewer tools are available to growers. So I think this is a really important area to learn more. I went out, checked things out, and we learned quite a lot. So results so far, we visited Ophelia's farm, and we checked on several different varieties that seemed to be symptomatic with agave mites, checking, okay, are these actually agave mites or is this something else? We also brought back several examples of agave tequiliana to try to inoculate them with agave mites and see if they would actually serve as hosts for agave mites. And basically what we found is that agave durangensis, which is grown for spirits production, had very clear agave mite symptoms. This is where a lot of these pictures come from, and this is also a zoomed in picture that I took on agave durangensis, where there's that agave mite pictured right there. So this particular variety had clear agave mite symptoms and was definitely suffering a little bit of damage. Symptoms were widespread on new plants that hadn't been planted yet, and also on plants that had already been planted out in the field. What do we find for tequiliana? We brought those back to the lab. We wrapped them up in parafilm. We put the agave mites on them to try to see if the agave mites would actually live on these plants. We inoculated 9 plants with agave mites. 5 had been grown from tissue culture, 4 that had not. I don't think it's ultimately mattered. There was no difference between these two, but it does happen both on tissue culture plants and on plants that are grown in more traditional ways. And basically, we found agave mites present on these tequila plants after 3 weeks. They weren't high populations, but every single one that we had inoculated still had mites and they still had eggs, indicating that yes, the agave mites were feeding on them and they were reproducing on these plants. And then we let them sit for months and we found that the mites and the eggs were still present even 116 days later. So months later, every one of those plants still had agave mites on them that we sampled. So here's a little table that shows which of the plants had agave mites, which ones had eggs. So at 21 to 28 days later, and then at 116 days later. This is the average number of agave mites per plant and the average number of eggs per plant. And these are pretty low populations. It's, less than 10 mites per plant and about 6 eggs per plant. Not too many. 116 days later, the population is still small, but it has grown. So there's about 18 mites on average per plant and about 8 eggs per plant. So all is to say that it's unlikely that agave mites live well on agave tequiliana. I don't think they don't affect the plant very strongly, or that tequiliana is a particularly good host for them. But more research is needed, and it definitely indicates that yes, even this very, very popular tequila variety does actually have the mites on it, and they can potentially grow and potentially could cause problems on this plant. However, because we haven't heard anything about this from growers out in the field, my suspicion is that agave mites don't do well, even if they can grow on agave tequiliana. But when we were doing that field survey of different varieties, There are plants that are strongly affected, and we'll show you which ones now and just how strongly. So remember, Durangensis was heavily affected, but we checked numerous different agave varieties that were growing out in the field. So here are the different varieties that we're looking at. Over on this left-hand side, we have Proto-Americana, Mapisaga, Atrovirens, and then we had 2 blocks of Durangensis. So one section that was a little bit closer to the rest of the plants, then another one that was a little bit further away. And basically, as we were going through, we were quantifying agave mite symptoms. So did the plant have no symptoms? Did it have only mild symptoms? Did it have moderate symptoms where, oh yeah, it was very clear it had mites? And then we're getting into severe symptoms and replace plant. Basically, severe symptoms was this plant is probably going to die from the agave mite infestation, and replace plant basically means that the plant was already dead from the agave mite infestation, and at that point, you should be getting rid of it because there was absolutely no hope from that. And then additionally, we had one other category, which is that some of the plants seem to have had an infestation and then grown out of it. So we're like, they had minor to moderate symptoms that were old, but the new leaves weren't particularly affected. So this is what we're looking at. For these first 3 varieties, we had hundreds of plants that we were checking, and basically none of them had moderate, severe, or anything higher type of symptoms. Only a few of them were maybes for mild symptoms. And in basically all of these cases, I'm pretty sure none of the plants actually had agave mites. They just had slight, lesions or physical damage that looked a little bit like agave mite damage. So I don't think any of these had agave mite infestations. For Durangensis, the story is very different. So some of them definitely had no symptoms, but as you can see for block 1, a lot of them had mild symptoms and a lot of them had moderate symptoms. And then for block 2, which was much bigger, while there were quite a few that had no symptoms, many had mild symptoms, many had moderate symptoms, and a lot had severe symptoms, and several were again completely dead at that point. So basically around half of the plants we checked had mild symptoms all the way up to you should replace this plant. And beyond that, so the agave durangensis we had was the only variety we checked that had mites. But this grower, Ophalia, had actually already removed 52 plants from the durangensis block 2. So basically that means that replaced plant was probably up at like 61 of them. She had removed these almost certainly because they had severe agave mites and had died from those already. So all is to say is that in the correct circumstance with, say, agave durangensis, you could get maybe 50% yield loss from agave mites if they sort of start going out of hand and really start growing. So it really is important to check your plants early for mites, because if they're there and if they're present for a while, they certainly can cause high losses in susceptible varieties. And again, for right now, the only one that we're aware of is durangensis that is commonly grown, but there certainly are other ones that could be there. Atrovirens, Mapisaga, and Proto-Americana seem unlikely to be heavily affected by the mites. Okay, so as a summary of all of this, what should growers do? What should agave spirits growers do when it comes to agave mites? First off, I would highly recommend you check any new plants that come in, especially say if you have agave durangensis, and at least look for symptoms on the plant. If you want to get more involved, then using a black light and or scopes, cutting open the plants, looking for those agave mites, that will definitely be useful and I think will be worth your time, because you could be bringing in new plants, some of them which have agave mites, so they simply don't have symptoms, and you won't know about it unless you cut up a few of them and sacrifice a few. So that certainly could be worth your time. As much as possible, of course, favor varieties that aren't affected. As far as we know, agave durangensis is the main susceptible variety. We're certainly going to be finding more, I'm sure, so be aware of that. Prevention and sanitation are critical. So if you have plants out there that are infested, I highly recommend getting rid of them early, especially if they're young, to prevent it from spreading further and becoming a bigger problem. So roguing your plants, getting rid of them is unfortunately probably one of the best things to do. Preventative miticides are probably some of the best options, but there is a catch to this, which is that it's very unclear what exactly is actually labeled for use in agaves that are grown for spirits production, because this isn't a very big thing here in the United States. There are some options that you can use that are basically used, labeled for use in almost anything. Say Tetrakerb Max, for example, is like a mixture of different oils that's labeled for use in basically everything and is also organic. You could probably use something like that, but we know that one doesn't work curatively, and we don't know if it works preventatively. Things like sulfur can be organic. I don't think they're labeled for use in spirits agave, but that is definitely a thing to be looking into and to try to be aware of, because again, as we found, sulfur was very effective. It can be organic. So we should look and see if we can't either get that labeled for use or find some situations where it would be labeled for use in agave spirits production. The last thing and the most important thing, because there's still so much we don't know, and we're still kind of, in the Wild West when it comes to dealing with agave mites and spirits agave, is to be your own scientist. Keep track of things yourself, take good notes, share information between growers and each other. All of this is really, really important because that's how we're going to generate more knowledge at this point. It's going to take a while to get more official results for a lot of different stuff. So sharing information and keeping track of things and, again, really trying to be your own scientist and figure things out will be very, very important. So those are all the different things that I would highly suggest everyone do. And with that, there's one last thing is, of course, stay tuned for future results. You will learn more about this. We're already doing more work. We're already figuring out more different agave varieties and species that are being affected. We're going to find out more in the near future. So stay tuned for future results, and hopefully we get some more good news coming your way. With all of that being said, there's a last thing I want to talk about, which I don't really like talking about, but unfortunately is a very big reality, especially for more specialty crops nowadays. And that's talking a little bit about the future of science and also the future of science at UC-A&R. I know a number of you on this call work at UC-A&R or work with UC, so I'm kind of preaching to the choir to a number of you, but I think this is really important. And I also think it's important to note that the following are my own views and do not necessarily represent those of UC-A&R. But this is a very interesting situation we find ourselves in, where the current administration has very significantly changed public science, defunding many, many different things. Ag has been somewhat shielded, but we still are feeling many effects from this. I've had colleagues at USDA who have been fired. We've had collaborations with all of this that have stopped. And there really is huge uncertainty about future funding opportunities, especially for things like small scale crops, things that don't have the capability to have, commodity boards that have a lot of money behind them. It makes it a lot more difficult when federal funding is curtailed. Research, for example, in this presentation was federally funded. A lot of this is done through IR4 funding, testing a number of these different miticides. And not only were we testing those miticides with this money, we were also doing a whole bunch of that basic research that's helpful across all different types of growers. The Trump administration, of course, is also clashing with universities, as I'm sure all of you are aware, pulling funding from universities. Harvard is a good example of that, where they are now currently in a lawsuit against the Trump administration. For UC-A&R, only about 10% of our funding is federal, but a lot of it also comes from grants. And of course, our tax-exempt status also might be threatened, depending on what the situation is and what exactly happens. A lot can change in the next few months. And one of the last things, I think this is especially important, especially with a crop like this, that sort of has such a cross-border history and connection. Many academics and students working within UC-A&R are not U.S. citizens. They're legal permanent residents, but they don't have the same protections that citizenship provides. And that's very important for us to be aware of. As an example, not that long ago, we received an email making sure that we knew what our rights were and establishing who to contact if ICE or DHS were to show up to our workplace. So all this to say that this truly is affecting our work, and it truly is affecting our ability to be good scientists. In my view, I think science should always be nonpartisan. It should never be used to just bolster one party or another, or have results hidden to try to bolster up one party or a different one. But science is not apolitical. It can't be, because all these different things can always be made political, and clearly they are being made political right now. It is my very strong view that you can't be neutral by staying silent about this, because staying silent is still saying something. It is saying that everything is okay, that it is still working and it's still functioning. As we can see, it really isn't very well at this point. So if this is important to you, I highly encourage you to speak out, to pressure elected officials, and to certainly work amongst yourselves and provide that mutual support network. And with that, I see there's a lot of questions and comments in the chat. I'd be happy to answer any of them. My email is down below at egmiddleton@ucanr.edu, and I'd be happy to answer any questions you have. Thank you.
Thank you, Eric, and definitely in agreement with your statement that we cannot be silent. That, yeah, we've been affected, and that, yeah, this is very unfortunate that we're in this condition, but that we have to respond. Hey, if you go back to your summary, I'll go back to the questions, but in the summary you have, that one there. There is, I think, some arthropods that also show to be very preventive, right?
So Neoceolus californicus could be. Yeah, I'm hesitant to put it in this category of yes, it will work. again, because I want to do that additional experiment. But you
Okay.
are correct that there is a decent chance that Neostilus californicus could also be used preventatively. So that's
And
another thing to keep in mind for sure.
yeah, and I mean, yeah, this is like, yes, you're saying this is research in progress. I was
Mhm.
just kind of remembering because I mean, I know that many of the people listening might be on the organic, not so much on the chemical part.
Yes.
But as you can say, they can be sulfur that is organic. Now, there are a lot of good questions. I want to go back to, so we have Steve's. And basically, Steve Sanchez says, have you ever tested high saponin agaves for mites host potential? Saponin serve as a biodefense, seem they would be more resistant.
Yes. So we haven't done any of the different testing for like, host resistance or like ways to sort of harden the agave and make sure that they are more host resistant. I imagine that that could be the case. And I think that'd be a very interesting thing to test. Additionally, one of the things is that most of the different varieties we've been looking at have been those present in nurseries where they're grown for ornamental production, where they're a little bit more stressed. So I imagine those plants are also kind of a little weaker. So we haven't tested that. I think there likely is something to it, but we simply don't know at this point. And it's definitely something we'd like to look into more. Because of course, if you can, make sure your plant is healthy and it's producing these defensive compounds to begin with, that'd be great, and you wouldn't need to worry about mites. But we don't know if that's the case right now, and it is something we need to do more research into.
Okay, thank you. And then from Mahesh, there are actually two questions here. So one, are there any level of symptoms for extreme climate? So have you seen agaves showing symptoms of any specific stress due to extreme climate?
Hmm. Um, I guess is the question saying that like if it's in, more drought stress conditions, is it more likely to get mites or something like that?
Can you can you
Yeah. So the answer seems to kind of be the opposite, weirdly. We haven't tested this again, but we have had situations where we have like a bunch of agave plants that are kind of sitting out at the back of our office that we've tried to inoculate and use for different experiments where we just build up a population of mites on these. Sometimes they're just sitting out there in the sun and they won't be watered as much as we should, and they're just kind of dying and languishing. And it seems like the mite populations will also die off with them. Sometimes a couple of them will survive, but it seems like oftentimes plants that are, being decently well taken care of, certainly getting enough water, enough fertilizer, and things like that, will still be in the mites, and the mite populations will survive a little bit better on them. That's all anecdotal, but I do think that there's nothing that really indicates that the mites will be targeting specifically stressed out plants. It seems like they will be going after a number of different plants that appear to be relatively healthy otherwise.
Mhm. Okay. From Nick Torres, is there any evidence of other ornamental plants having agave mites, like cactus or succulents?
Yeah, so do other ornamentals have agave mites? So mangaves probably do, but other different things like other cacti or aloes or stuff like that, no. They have their own mites that are different. See, aloes, for example, have aloe mites, which cause very different symptoms, but they look basically the same as agave mites. So no, agave mites are almost certainly host specific to agaves and mangaves, but some of these other succulents will have their own issues that are separate.
Okay, we have also another question from Ivan Ojeda. So how was the sulfur applied? Was there a dilution process?
Yes, so the sulfur was it's basically a wettable powder that's mixed up with water and then applied just the way like lots of other sprays are as a foliar application. I don't remember the exact dilution of it. If you're interested, shoot me an email and I can send you like the exact amounts. I don't remember what it was. It's like max label rate, though, basically for the sulfur that was used as a fungicide. So I can't remember off the top of my head. It's decently high, but it's like, it's basically was listed on the label.
Yeah, and sorry, I kept thinking on the species that can be affected by agave mites, and you mentioned Durangensis. That one was a big. They really liked the Durangensis. They didn't like others, I'm
Correct.
thinking perhaps more of my palate experience that mezcales from different species have different flavors, and I think they come from that um their composition, right? Their cells
Mhm.
might be different, and those the mites may like one mezcal of Durangensis more than another one from other species. Anyways,
Definitely.
um, but yeah, I was just kind of surprised. One other question that I have, sorry, I'm the geek or the scientist is coming here. So I saw that you had, so on the treatment, when you were trying to treat it and then when you were trying to prevent it,
Yep.
I think when they were already infested and you do the treatments for trying to cure them,
Yep.
it is really difficult to say like, okay, I'm gonna have a 50 mites in each plant, right? So you were trying to infest it and then later see if there was a difference in the treatment compared to control. I think
Yes.
sometimes control was above, below, but that is because you cannot say like, okay, these plants, 50 mites, go my, have at it, my friends, right? That's why
Exactly.
you were,
Yeah. So that's one of the things that's so tricky, and that's why I wanted to do another experiment with the Neocoelus californicus, is because there's so much variability in how well our infestations take. And I think we've kindly got to a point where we're pretty consistent, but even then it's so hard. And because the mites, you can't count them unless you kill the plant, it's really hard then because you can't like count them beforehand and make sure you're doing well. You just have to kill the plant and then it's done. So it's tricky.
I was thinking the same, and you know that's what I was, because in many of your graphs, I mean the control was below, above, and sometimes, so it seems that sometimes the count, the error bars, they were really large, and
Yes.
not necessarily because they were errors, it's because the counting is, some plants the mites really have at it, some other ones they were just like, meh, I don't think I'm gonna get in here.
Exactly. So there were some situations where, one of the plants had like 100 something mites and the other one had three. And so that means, yeah, we have huge error bars because of that. But one of the things that was kind of neat is that even for all these different myosides we tested, there was basically always at least one plant that had that like about 100 mites. And so that was useful for us because then we're like, okay, so even though there is a lot of this error, we do know that, you know, there's instances where it's not working at all. So,
Right. And I was also thinking, so do these mites, it looks like they come with the plants when they bought it, right? Or is it native here in California, the mites?
we don't really know. They definitely are in the landscape occasionally around here, but as to where exactly the mite is native to, it's unclear. So it seems likely that, like say when Ophelia is bringing some of these plants in, that they did have the mites on them to begin with. Um, but it's we don't know. But that's another thing is that like, why is it that there's so many growers down in Mexico who are producing Durangensis who probably don't seem to be having this issue at all? Is because they simply don't have the mites? Is there something else that they're doing that's is making it so the mites aren't a problem? We don't know that. And just like looking through the literature, there's nothing. It's just not there. So it's
Yeah,
it's
as you're saying, it is really difficult. I think also just want to make sure that we hit it home, that in this one is really prevention, like, and
yes.
it is prevention at all times. So if you're buying the plants, if you're buying a bunch of plants and you invested a good amount of money, it's really worth it to invest $90 more to have a lamp where you can check it, right? For me,
Yes.
that was like, and yes, you're gonna have a good amount of time on your bench inspecting all the plants one by one, no way around it. Like,
Yeah.
you want to better catch it first there. So
Mhm.
if you plant it, then it will not spread out.
Yes. Now, one thing to be clear about, I completely agree with that. Just the one thing is that even using the black light, you have to kill the plant to check for symptoms
Ah,
because you
okay.
rip it apart and look like at the base of leaves and in the core. So I would say like if you're getting, 100 new plants in, like take five or something and check those and make sure you don't have, mites. present. Again, that's a sacrifice. It takes time, but especially if you have a susceptible variety, it's definitely worth doing as opposed to putting it out there. Because say with Ophelia, again, she put a bunch of these plants out and was getting like 50% yield loss. That's a huge amount of effort and a huge amount of time.
Yeah, yeah, and again, it is time and it is dollars, but you have to be preventive. And then the other one is some of the treatments. I mean, we saw that the Arthropodus californicus, that one, like, still to be determined. Keep it just kind of on the back of your mind. We have the sulfur that it seems that is working, and that is just prevention. Like that one we need to get going. And once you have it, then if you have mites, then identify, take the plants, try to isolate it, get rid of them because it will spread out. Actually, you have, Eric, a nice proposition by Steven. If you ever want some saponin to infect and test, that you shouldn't let him know. I can send you his information.
Yes, if you could, that'd be great because that'd be really fun. It's really easy to try inoculating some with mites and see. And yeah, we can give it a shot and see. That'd be very interesting to know. If there's any way to have like the same species, I don't know if this is the case with like high saponins and then low saponins, like that'd be perfect. But even if just sending, some of those, that'd be great because I'd love to test it.
And I mean, Steve is going to the prevention really from the root, which is basically we can hybridize and then make it bulletproof. So that will be prevention right at the beginning.
Exactly. And that would be ideal because, of course, then you don't have to spray things. You don't have to do all this other stuff. Like that would be the ideal situation. So, yeah.
Anyways, I think we're good on time. I would like to thank everyone who joined. First and most importantly, Eric, thanks a lot. That was also a great presentation. I really like the scientific content, and at the end, I think, like, yes, we should speak up, including myself. Yeah, things are no longer fun, and we are here for the benefit of everyone, regardless of the color, regardless of this is for everyone. So anyways, thank you for joining. I will